Welcome and thank you for joining us for the 5th Agro Lunch and Learn webinar of 2023. We are delighted to have Eurofins Agroscience Services as the generous sponsor of this webinar series. The Agro Division extends our heartfelt appreciation to Eurofins for their continuous support. My name is Solito Sumulong, Director of Regulatory Services at AgriThority, and we're based in Kansas City. It's my privilege to serve as the moderator for today's webinar. Use the Q&A tool to submit your questions and comments and make the most out of your attendance today. Our team will actively monitor and respond during the presentation. Before introducing our speaker, let me emphasize the immense benefits of joining Agro. Enhance your science, expand your scientific network, and support your professional development. Visit agrodiv.org and click on Become a Member to join as an affiliate for only $20 a year, regardless of your membership status with the American Chemical Society. For the next fall meeting in Denver, we are planning exciting agro programming in line with the meeting theme, Elevating Chemistry. The organizing team reminds organizers that November 2nd is the call for abstract deadline. For any programming inquiries, please reach out to E.J. Gallo's James Foster, who's the program chair for Denver 2024. Now, I'm proud to introduce our speakers today. Dr. Henry Krueger is the VP of Science and Regulatory Affairs at Eurofins Agroscience Services, which is formerly Wildlife International in Easton, Maryland, and has been working in regulatory ecotoxicology in both microbial and conventional agrotechnology for 35 years. Recently, he delivered a talk to OECD in Paris on the challenges of standard aquatic toxicity studies with microbial products and moderated a BPIA panel discussion on anything new with global regulation of biological products. He directed and managed business, finances, technical programming, and development for aquatic plant and insect toxicology in Easton for 25 years. His background was established in both aquatic and terrestrial ecotoxicology early in his career at Wildlife International as a 10-year director of field programs. Activities included mesocosms, terrestrial and aquatic field studies, as well as plant studies conducted in greenhouses. He has an MS from Central Michigan University and a PhD from the University of Wyoming, where he works at the U.S. Fish and Wildlife Cooperative Research Unit. His particular interests and experience are in algae, honeybee, and bumblebee lab testing, fish bioconcentration, fish and frog endocrine disruption, invertebrate sediment testing, plant injury scoring, and the development of new testing methods. Now, Sarah McCoy is a study director and team lead of the Biologics department at Eurofins Agroscience Services in Easton, Maryland. She has a master's degree in molecular biology and genetics from the University of Delaware, and a research background in biomarker discovery with extensive experience in molecular biology assays, including RNA sequencing, qPCR, and protein quantification methods. Sarah has worked at Eurofins for 5 years, developing experience in aquatic toxicology. She and her team are working to extend GLP testing capabilities on biological pesticide products for support of ecotoxicology studies and microbial product characterization. So, with no further ado, I'll hand it off to Hank Krueger.
Well, good afternoon to everyone. Sarah and I are going to be talking today about biological product registration and testing, and we're going to do an emphasis on aquatic toxicity testing. When we talk about biological products, there's just a need for some basic definitions. It covers a lot of different areas, and I think what I wanted to talk about to begin with is just getting kind of common terminology. These biological products include non-microbials, biostimulants, microbial pesticides, biopesticides, biocontrol agents, and macroorganisms. Fortunately, there's been a chart that's been put together by the Dunham Trimmer organization. I use this almost every day, it seems, but I think they've really roadmapped all these different products and how to look at them. To begin with, they break them down into two different major categories, which are biostimulants and biocontrol agents. And if we looked at the left side of this chart, there's a further division. And now we start getting into the granularity or maybe into the weeds a little more, where the biostimulants can be microbials or non-microbials. For the microbials, they're usually bacteria or fungi, and they're going to just increase the the efficiency of nutrient uptake in plants and promote growth. On the non-microbial side, you have things like the plant seaweed extracts, amino acids, organic acids, and inorganic compounds. And when we start looking at the regulatory aspects of this, the biostimulants are sort of a special category, and they never really fit with the existing. It's newer technology, and with the existing terminology, didn't fit. Well, right now, there's bills pending in the House and Senate with sort of the same language that are redefining what a plant biostimulant is, and they exempt it from FIFRA, which would be a battery of tests. The only exception would be if a product claims to have pesticidal activity, insecticide, herbicide, those types of things. And I think it's worth just kind of reading this definition. The term plant biostimulant means a substance, microorganism, or mixture thereof that when applied to seeds, plants, the rhizosphere, soil, or other growth media, act to support a plant's natural processes independently of the biostimulant's nutrient content. But more importantly, it's thereby improving the nutrient availability, uptake, or use efficiency, tolerance to abiotic stress, drought, things of that nature, and consequent growth, development, quality, or yield. Excuse me. What's great about Dunham and Trimmer, they look at these kinds of markets, and what you find is that the majority of biostimulants are in the nitrogen fixing bacteria. They make up the largest segment, but the fastest growing segment, and to me is pretty exciting, is it's for non-leguminous crops. And when you think about that, they're turning corn into nitrogen fixing kinds of plants, or assisting in that area of nutrient uptake. It reduces nitrogen you're putting on the field and runoff and those kinds of things. On the other side of things, you end up on the chart was biocontrols. Those subdivide into the biopesticides and macroorganisms. I'll start with the macroorganisms because most of you are probably familiar with these. These are things like ladybird beetles, lacewings. They might be used in integrated pest management and are used in greenhouses, things like that to control insect pests. On the biopesticide side of things, you end up with biochemicals and microbials. The biochemicals are usually plant extracts, organic acids, plant growth regulators, or semiochemicals. And the semiochemicals are things like pheromones. On the microbial side, you have bacteria, fungi, protozoans, viruses, yeast, and others. And if we look more on in terms of regulation, just kind of a quick summary, the biostimulants are safer products. They're usually natural in origin, and they're not regulated under FIFRA, but they're regulated by states. And the current efforts, if you get it out of FIFRA, is to come up with some common templates for the states so you don't have to have 50 separate types of submissions. On the biocontrol product side, the macroorganisms are regulated by USDA. And under APHIS, basically, if that organism, the ladybird beetle or whatever, is native to an area, there's no problem. You don't even need a permit. Permitting is required when you bring something in from a foreign country or something from outside the range of that species. Now, the biopesticides are regulated under FIFRA and therefore require the testing battery of ecotoxicology. Like we talked about before, these are biochemicals and microbial products. And today we're going to focus more on the microbial because those the biochemicals kind of fall under conventional testing. And we're going to be emphasizing aquatic tox tests. And most of the aquatic tox tests for registration of the MCPAs are Daphnia and fathead minnow or other fish species. We have a large body of knowledge for conventional testing that can be applied to these kind of materials. However, our level of experience with microbial products is not near the same as we have with chemicals. There's major differences between testing a chemical and microbe, as you can pretty well decide on your own. We need better guidance and acceptance criteria for tests. And we also need to, in my opinion, really have experimentally derived data on how it's going to react in the test system prior to testing, or if we even need to test. We need to consider routes of exposure and risk evaluations. And to me, this is a new thing. It's no different than like conventional chemicals. We need to re-examine our assumptions and make necessary modifications on how we evaluate risk with these products. Well, what are the differences between conventional chemicals and MCPAs? Microbes are particles. They're cells, spores, cysts in suspension in water. And while chemicals are water soluble and form really true test solutions. The testing method for chemicals avoid testing particles. I spent most of my life in aquatic tox trying to keep particles out of water. Water solubility sets the upper limit. You don't want it to precipitate. However, with microbes, now you're putting particles in water. It's a game changer. It really makes things different. Additionally, the microbes are going to react both in an abiotic and biotic processes. They're going to grow. They're going to multiply in your test systems. They're going to interact with other microbes in your test system. And so we really need to develop better testing methodologies to design better tests. What are our typical problems when we do tests with microbes in aquatic test systems? Poor visibility of the test solutions. They'll turn white, they'll turn different colors and be very opaque. So it's hard to really see your fish or Daphnia in that system. There's difficulties of spores or cysts in water. Are they going to suspend themselves? Are they going to sink? Are they going to float? And that goes into, with all tests, the requirements are stability and a homogeneity of the test material. And with these kinds of materials, we need better definitions or rules of how that we define those terms. Typical problems you see with fish is clogging of gills. When we go to Daphnia, they're filter feeders, so particles interfere with the efficiency of feeding. And there are some works being done now how to increase the amount of food or that to counteract those kinds of decreases in efficiency. One of the things that's always tough about these tests is separating out physical toxicity from general toxicity. In other words, if I clog the gills, it's really not a toxic response. I just suffocated the fish. And also, how do you tease out toxicity, infection, or pathogenicity in, say, a Daphnia? So, we still need some work on how we define these terms. I'd like to spend a little time in how important it is to have the right team of people for testing. And, to me, it's in the companies, it's also in the regulatory reviewers of really having a command of what's happening. The MCBs are typically safer alternatives, and therefore we need to consider how it's used, what the application methods are, and what the environmental conditions are necessary for survival to determine even if testing is necessary. We need to determine realistic exposure assessments, which I'll go into more detail. And we got to develop good microbial methods, just like we do with chemistry. We need quantification of the test concentrations, whether it's CFUs or other kind of numerical counts, biomass, whatever. And one way to get some of that information prior to testing is really important. So Sarah's going to go into more details how mixing trials are going to tell us how it's going to react in water, how we can prepare our test stocks, and also use that information to assess environmental exposures. What's going to happen when this stuff hits water? Well, as I mentioned before, I want to really kind of talk about how important it is to have the right type of people. When I started, all you needed in aquatic toxicology was a biologist and a chemist. And, that was sort of the level. Some people talk about it as kill them and count them kind of toxicology. It was mostly survival, acute type tests. However, if we look at how toxicology has advanced, and endocrine disruption, I think, was a game changer because it really complicates the life of the study director because now you're dealing with several levels of biological organization. They're still going to have to work with the chemist, but now they're going to have some idea of physiology, cell biology, endocrinology. There's more endpoints. They're going to have to work with statisticians. They're also going to have to work with histopathologists as well as molecular biologists to cover all those endpoints. When we move to microbial testing, you can kind of replace the chemist, the role of the chemist, with the microbiologist. And I think this is really, you still have these different biological levels of organization that you need to consider. But between the microbiologist and also I'd maybe highlight the molecular kind of biologist geneticist, it opens a whole new level of like going down the rabbit hole of bioinformatics, gene sequencing, and additional PCR analysis. And I think that's, the technology is changing and, we just need to keep up with it. Well, I'd like to talk about a little bit how we evaluate risk, because hazard, I think we all understand. That's just the inherent toxicity of a chemical or any kind of test material. It's an LC50. It's an EC50, it's a NOAC. On the exposure side, we're dealing with how much of the chemicals actually present in the environment, and then how much of that organism is really going to come in contact with it and what that uptake would be. To me, we really need to focus on is that exposure component. Now, when we look at conventional chemicals, we really got a good understanding of what happens when you put a chemical out in a field. We have the field trials we need to collect information. We have physical chemical data. We can plug those into models and get pretty good estimates of what Europe usually calls a predicted environmental concentration, or in the US, estimated environmental concentrations. Now, typically, what regulators will do is look at some kind of ratio between that environmental concentration, that exposure, and toxicity. And this is in the form of usually risk quotients, where you take the the EEC, expected environmental concentration, and divide it by the most sensitive LC50 measurement or NOAC level that you have. And when we look at aquatic toxicology, for aquatic animals, if you look at the presumption of risk, we have acute and chronic, but the acutes are high risk, restricted use, and endangered species. And what you see in the RQ values is, again, the importance of the expected environmental concentration divided by that LC50. For chronic risk, it's the EEC over the NOAC. And when you look at that LOC, which is a level of concern, when the NOAC exceeds the EEC, that's a level of concern. When you get to the acute high risk, it's really the LC50 has to be greater than 1/2 the EEC. For restricted use, it's 1/10, and for endangered species, it's 1/20. Well, as you can see, it's going to be very important in considering routes of exposure when you determine EECs. The MCPAs are not as straightforward and predictables as chemicals. What does drift mean for microbial products? What does runoff mean? And what are the mechanisms for that to occur? And, how are we doing these applications? Are we applying directly to water in the field? Because that's all going to figure out how much is going to get into water. And I think, again, we need to develop better exposure models and use field residue studies or some other surrogate to accurately predict what the environmental concentrations are going to be. Another issue that comes into play is how high do we need to test? For conventional testing, it's pretty much driven by water solubility is a limit, or if solubility isn't an issue, it's 100 milligrams per liter for pesticides, 1,000 mg per liter for industrial chemicals. And for endocrine, we use a maximum tolerated dose, which is 1/10 or 1/3 the LC50, depending on what you're looking with the reference you're using. For most of the MCPA products, it's a two-tiered evaluation, and the upper tier is a limit test, means you just test a single concentration. And that concentration is defined as the maximum hazard concentration. And I want to talk a little bit of how that is calculated. both USP, the EPA, Canada, and Europe kind of use these kinds of metrics. For water, it's 10 to the 6 units per mil, or 1,000 times the expected microorganism concentration in the aqueous environments, whichever is greater and readily attainable. Typically, it's the 1,000 times. And that's calculated by just taking the maximum, application rate and, making the assumption it's directly applied over a 6-inch layer of water, or 15 centimeters, depending on units that you're using. For food, it's 100 times, but again, a similar calculation. And if we move on, we can see there are exceptions. And one of the things that I think is very important if you have requirements for testing is to make sure you're interacting with the agencies that you need to. Canada has a formal pre-notification consultation, and I know EPA will listen. I think you need data-based arguments, but you can reduce that from 1,000 times, kind of like solubility. How much can I get in? What's a reasonable concentration to be testing? And I think you have to make a kind of weight of evidence argument, including how much is expected in the environment. And I've got to ask, do these MHC values make sense? These were developed at the time guidelines were really coming out in the mid-90s. And that was a time when we were really focusing on worst case scenarios. And I think, with probabilistic risk assessment, all these other type of things, we can look at this differently. Again, just to illustrate kind of how worst case this is, you take a maximum application rate. It really doesn't matter what the surface area is because you're developing a concentration here. So you determine the amount that's going on in that area and look at the depth, 6 inches or 15 centimeters. It's not taking into effect, does it float? Does it sink? What's going to happen when it hits biologically active water, when the wind's blowing, all those kinds of things, or what occurrence, if it's a stream or river. Factors to consider in this kind of exposure assessment is what's the intended use of the product? Are you putting it on a field, or are you doing a direct water application for mosquito control or something of that nature? And that makes a big difference. And I think really when you put it on a field, how is that MCPA even going to get into off-field bodies of water? Well, a couple things to consider is the formulation, because wettable powders will probably behave different than liquid formulations and granules. The application method to me which is really key. And as you see, I'm going to be talking about decreasing risk as we move down this list. You have direct overspray over the water if the product's meant to do that. But then you have a broadcast on a field. That could be a spray boom, an air blast spray, or a plane. Then you have in-furrow application. And then you have a seed coating. And I think the risk has to factor in. If I'm just putting this in as in-furrow, or I'm putting on a seed coating, how is that going to get into a body of water? And, we've got to have, a pretty good understanding of when testing is necessary and how much we need to actually testing in terms of the concentration. The other wild card too is that MCPA is going to go into a microbial community, and it's going to be both on and off target. And one of the questions is that MCPA even going to, survive in that off-target microbial community or a microbial community in water, or is it just going to dormant, that type of thing. When we consider routes of exposure, the GLPs are pretty, specific in that you need to designate the route of administration and reasons for its choice when you test. Now, for conventional chemicals, it's kind of a no-brainer because most of these materials are going to have some type of water solubility, and some of concentration is going to need to be tested. With the MCPAs, I'm not sure it's quite the same whether we have a good understanding of the aquatic roots or what's going to happen when it hits an aquatic test system. Again, how are these things going to get there? How do you define drift with MCPAs? How do you find runoff into streams, ponds, lakes, or mechanisms for runoff? Evaporation, I don't think you're going to have to worry about. That's more for conventional chemicals. And you always have the case where it's a herbicide control of aquatic weeds or mosquitoes, where you'd have direct applications to water. And the chemicals have models and residue studies to accurately predict the environmental concentration. I think these need to be developed for these MCPAs as well. Kind of the final topic I want to hit before turning it over to Sarah is the decision whether you're going to test the kind of an active ingredient versus formulation. Usually what comes to us in the lab is a stable powder or a broth with an MCPA inoculum, and that's what we're going to mix and form our test solutions with. Formulations are typically not a good idea. You have the option with regulatory to do both. There are some formulations that are unique to microorganisms that can be used. But if we look at how these materials are delivered and what amendments are put into them, this comes from Varsha Barty and Shahida Ibrahim, where they look at the different types of amendments added to MCPAs to either give them shelf life or be able to apply them. And if you look at the examples, none of these things really behave well in water. You've got molasses, you've got vegetable oil, you've got kaolite clay. I like organic carriers for grain flours. What happens when you put water and flour or cornstarch together? You make gravy. And and putting fish in gravy just doesn't make a lot of sense to me. And Sarah, I'll turn it over to you.
All right, thank you, Hank. I'm going to talk a little bit about, with all of this information and improving models for exposure and hazard assessment, what can we do to improve testing and what tools are at our disposal? So as Hank discussed, a lot of groups are working towards improving guidance surrounding biological product testing, and the exposure models are evolving. We have some clarity on what tests need to be run and what data is required for submission. However, there are practical and technical implications to testing these products that need to be considered. As a CRO, we feel that it's important to standardize our approach to testing these compounds. And for today's purpose, I'm going to use aquatic testing as an example, as Hank did, but the same considerations can be applied to other test matrices and other organisms. So ahead of ecotoxicology testing with conventional chemicals, we acquire a lot of information that helps us predict how the test substance is going to behave in the test system. Not only do we have sufficient models to estimate exposure, which help indicate what types of tests are needed, we also have information about the test chemical itself that helps dictate what the test system is going to look like and what the test conditions are. I.e., we have information on water solubility. We have some information on the degradation of the chemical. Is it photolysis or hydrolysis? If the chemical is volatile or not will change the test conditions. Where it's partitioning in the water, if it has potential to become a bioconcentration. And then also where it partitions, does it absorb to the test vessels, systems, tanks? All of that's going to change the way we set up the test system. How can we mimic this, and what kind of studies can we run to design, upfront studies that we can run to design MCPA testing? So how do we apply this thought process to testing MCPAs? Our approach is to model the preliminary testing after what is typically done for conventional chemicals. So in addition to the physical properties of the test substance and how it interacts with the test system, there are also additional biotic factors that come into play when you're talking about microbial products. So some of this information can come from a literature review, depending on how much prior work has been done on the microbial, but the remaining can come from carefully designed pre-testing trials. Bioinformatics can be a powerful tool. It can be used to make predictions about a microbial test substance. It can identify closely related microbial organisms. It can also identify what might be a closely related non-target organism to the organism target. It can identify metabolites of interest and predict pathogenicity. Viability is also a major difference, as Hank mentioned, between microbial products and conventional chemicals. When you have something that's a microbe, it's alive in the system, there's possibility for reproduction and growth as well as degradation. The environmental conditions of the test system are also important. They may predict the viability of the microbial, but they definitely will have an impact on how it behaves in the test system. You know, the environmental conditions are geared to be ideal to the organism you're testing and not the microbial itself. We also need to consider what microbial presence is in the test system itself. You can have some interaction between the microbe and the microbials that are already there. There can be contamination in the water or the matrix. You need to consider the bioload of the organisms and biofilms that may be present. In some instances, it's also important to consider the interaction of the microbial product with the matrix itself. I'm just going to give the example of, you know, royal jelly or some honeybee diet properties. When you're doing honeybee testing, they can actually be antimicrobial, and that should be considered when you're setting up your test conditions. So what sorts of pre-testing trials should we do? The behavior in water can predict what route of exposure is more likely. You want to assess if the microbial product floats, sinks, or stays suspended. This can be indicative of, you know, if it sinks, sediment exposure may be more likely. If it floats, there's a possibility of further degradation because of exposure to the UV radiation. And if it suspends if it stays in suspension, a water exposure may be more relevant in that case. When we're considering, you know, we have a microbial test substance that we're going to be testing in the lab, it's important to assess the microbial product alone. This can be in the form of conducting pre-testing plating trials. The upfront work with a test substance can include characterization. You can get information on purity or contamination. It also allows for the ability to gain experience with working with the test substance itself and to determine if, you know, what we're given by the sponsor as far as a C of A, do we get that same type of result in our hands? It's also important to consider the viability and stability of the test substance alone so that you can later compare it to how that looks in the test system. So we suggest conducting mixing trials with the matrix of interest to help determine how the test substance will behave in the system, which allows us to get information in order to design a better test. So we would assess viability, you know, does it survive? Does it reproduce in the test system? Is there settling time if it's going to settle to the bottom of the system? Stability, does it remain or does it degrade? Homogeneity becomes important when you're talking about analytical sampling or what the test organism is actually going to be exposed to. Is there a need for aeration? And if so, does that aeration have an effect on the microbial product? The turbidity, the time interval before test organisms are added to vessels, and then whether or not you have a need for filtration, either before adding this test substance to the system or before analytical sampling. You need to kind of have an understanding of the size and shape of the product in order to determine proper filtration conditions. And then it's important to assess your controls so that you can determine what you're testing. So how does this information that we gain from these pre-testing trials inform the test conditions? So for example, a 21-day Daphnia test or a 30-day fish test, that type of testing setup requires static renewal design. So stability information will inform whether or not those renewal intervals are appropriate, or if you have a set renewal interval, whether or not you can expect to see higher levels of the test substance at the end of that renewal. As I said, homogeneity information helps determine sampling methods. Do you need to go at multiple depths? Is mid-depth okay? Do you need to kind of go to the bottom of the system? If you're considering how the organism is going to be exposed to the test substance, does the settling information will help inform the initiation timing or when you're adding the organisms to the system. And then, you know, what types of controls do you need if if the test organism is interacting with a microbial product, you may want to consider an abiotic control to see what the system would do without organisms in there, or a formulation blank if you're trying to determine if there's, physical toxicity as opposed to from the test substance itself. From an overarching perspective, it's important to continually be aware of whether the testing conditions are relevant for the particular microbial product being tested. So we're not really making changes to the types of tests that we're running, but to keep that in mind as we're testing different types of products, it's important to remain aware. You can consider if the timing of the test is appropriate to determine the biological endpoints that you're looking for. Is it practical for the target organism? So if you need to wait a significant amount of time for possible infection or pathogenicity to occur, are you using organisms that can survive in a test system that long? Microbial products are typically very specific to the target organism, which means that there should be consideration whether or not the non-target organism in the testing is appropriate. So if you have, an insecticide that is specific to a particular type of insect, you may want to consider the non-target organism that is closely related to that insect. This choice is obviously dependent on, what the target organism is, the application method, and where that test substance is going to end up in the environment. We also want to consider whether we're assessing infectivity or pathogenicity. It would take time for infection to occur, and then it may or may not cause a pathogenic effect. So we'll keep that in mind, and then also consider our controls, whether we need an abiotic, attenuated control, formulation blank, and do we have the capability of providing adequate analytical quantification of this substance? So what specific controls are we talking about? A negative control is kind of an obvious answer. It shows that the test organisms are healthy throughout the test and that the test system maintains the environmental conditions. Positive controls are useful, especially in conventional chemistry, but generally are chosen based on specific mode of action and demonstrate that the test organisms are sensitive in their response. So we do that regularly in testing, but it may be difficult to determine the appropriate positive control for a microbial product. Abiotic controls are useful for monitoring the MCPA in the test system without the confounding factor of organisms and the bioload that the organisms may bring into the system. Non-infectious controls are useful to show that the observed response in the test is the result of the microbe being tested and not due to other substances in the test, and can help distinguish toxicity from pathogenicity. Inactivating microorganisms by autoclaving may not be the best methodology, so it's important to be aware of these limitations in non-infectious controls. Autoclaving would kill the cells, which then causes a solution that now has lyse contents of cells that may cause toxicity in itself. A better solution is possibly to use a formulation blank, as we talked about, but that might not always be available. Ideally, we would need a non-viable control that has dead or a dead form of the microorganism. So it's difficult from a practical standpoint to discern mode of action and what may be causing toxicity, but we feel that it's beneficial to remain aware of the different possibilities. So this reiterates the importance of proper controls and relevant test conditions. So for example, if the toxicity is due to pathogenicity, there's a lag time for infection that occurs versus if you're dealing with chemical toxicity or acute toxicity. And then keep in mind that we need to reconsider the bioavailability. So what is in the system that is causing this effect? So we want to get an accurate representation of, what the organism is being exposed to in the test system. So we need reliable and precise methodology to quantify that. The standard methodologies available are standard or viable plate count. It's an indirect measure of cell density that represents counts of only the live bacteria expressed in colony forming units. So you need to develop a method that allows you to count, in a range of 30 to 300 colonies, and this can be complicated by, other bacteria in the system. Spectrophotometric analysis is a measure of the absorbance of how much bacteria is in a system via optical density. It's an indirect reflection of the number of bacteria, but it doesn't account for, it doesn't discern between live and dead cells. It also doesn't discern between types of bacteria. So if you have contamination from the test system or, other microbes in the system, it's not going to really help you quantify your test substance itself. Flow cytometry will count single cells, and it's useful if you have a stain or some type of selection that can be applied to your test substance alone. You can also stain for viability in that type of method. Bioinformatics involves gene sequencing of the microbial to identify MCPAs, and it's used when there are no known culture methods for plate counting. qPCR can also be used as a confirmation of the microbial product, but it does not discern between viable and non-viable cells. So the main limitation to all of these methods is contamination. So we've talked a lot about the fact that you're putting these microbial products into a test system that has, its own bioload of microbials. It has organisms in there that also have a microbial bioload. So the presence of microbes other than the test substance is the largest barrier to selective quantification of an NCPA in ecotoxicological testing. It presents a challenge for each of the methodologies that we've talked about. The easiest method to use to distinguish between, contamination and a specific test substance is plating. It's needed to distinguish because you can tell by the morphology of the test item by comparing it to proper controls that allow you to see the background levels of microbes. So this is an example from an in-house plating trial courtesy of our microbiologist, Luke. So you can see how it can be difficult to discern between, the test substance you're looking for and what else is in the system. So other considerations you can take, or other things you can take into consideration are filtering, abiotic controls, which is all information that you can get from those pre-test trials we talked about. We need to ask ourselves what precision, accuracy, and reliability mean for MCPA quantification. For conventional chemicals, the acceptance criteria that's generally used is analytical recoveries between 70 and 120%, with a residual standard deviation of 20%. So that varies widely when considering, plating or different methodologies for microbial products. We need to be concerned with how we maintain consistency between labs and between different types of test materials that have different applications. Pre-test trials can help inform not only the test setup, but also, this analytical criteria. You can get some sort of understanding of what the test substance is going to look like on the analytical end and can allow you to have a consistent approach that will improve the reliability of the test methods. So ideas for future improvement. The path to improvement in our opinion is going to be experience and new technology. So some of these pre-test trials will allow you to gain experience and expertise in identifying the test substance. It'll improve consistency in the plating methodologies. The use of proper controls will always be important. So then you can utilize some of these advances in technologies. So automatic plate counting is continuously improving. You will be able to, will acquire the ability to set specific criteria for colony identification, which also, and most of them also have the added benefit of, storing images as you're counting the plates. So you could go back for confirmation later or have a discussion about what is a positive result and what isn't. Like I said, for some microbial substances, you can apply selective staining or immuno-based methods, which improves the use of flow cytometers. This specificity will become especially important when you begin to handle products that are a mixture of two microbial active ingredients. There's also some qPCR methodology that's being developed that takes viability into account. So it has some sort of, barrier blocking for free DNA before you conduct the qPCR method. So that would be improvement on specificity as well. So overall, combining the expertise and improvements in technology is going to be the sweet spot for improving these methods. Another consideration is the need for a validated method that would include needing to include proper quality control methods. We can use the current chemical procedures as a guide, such as the Senko validations. MCPA validation should include proper quality controls, such as matrix fortifications, abiotic reps, attenuated controls, and matrix blanks. We want to identify, what the acceptability criteria is going to be for microbial products. So comparable to analytical recoveries for conventional chemicals, that's something that needs to be developed. And then our recommended progression, when you're starting out working with a new test substance is to think of it in a way that's a standard approach. So we start with those pre-test plating trials or a method transfer of a sponsor supplied method. You can do some method development work if it's needed at that point. The method validation becomes important because it would be in your matrix of interest. So obviously it'd be water for aquatic testing, but also sucrose or bee diet solutions for honeybee tests, or those carriers that Hank spoke of for avian studies. The mixing trials themselves would help inform the test system design. Periodic verification of your test substance in the test system is useful to determine stability over time. Then you come to your test system design and your reliable analytical quantification. So in conclusion, these testing approaches for aquatic testing of MCPAs need to be updated and modernized, as well as standardized. The routes of exposure and estimated environmental concentrations need to be established. There is a need for better exposure models. New standards for the methods used to quantify exposures during tests need to be developed, and acceptance criteria for testing is needed. The pre-test trials should be run to help design tests and determine if further testing is needed based on the routes of exposure. So thank you for your time today, and both Hank and I are available to take any questions as needed.
Awesome. Thank you so much. Thank you so much, Hank. Well, it's time for questions. We had 5 in the queue. We invite the audience to continue filling up that queue, and we'll have all the questions available to the presenters afterward in order to take this to an offline conversation if necessary. So let's start up at the top. I know an early question was kind of a batch of 4. I'm trying to scroll up the queue here to access it. From Santos Garcia. What issues have you had in using microbials and algae studies? As in, have there been significant issues in doing automated cell counts, as there are issues in distinguishing between bacteria and algae? Or can the counters easily distinguish between them? Or do you default to using chemocytometers to manually do algae counts? Be specific there.
Yeah, well, I can speak from experience. We're lucky in that we really haven't been called on to do algae studies. But because it's just inherently going to be difficult. You've got algae cells in there, and then you're going to add some other, a microbial, say it's yeast or anything, it's going to grow. And so now, the criteria of the existing tests, you're not going to be able to hit those growth rates. You could tell viability, you're going to have to factor in what's an acceptable test at that point. I would start with pre-testing, putting the material in the algal media itself, and just determine, is it viable there? Or does it just stay as a dormant spore because it doesn't like being in water, because it's a soil kind of critter. It's so that it's a very difficult test, I think, to pull off. I don't think we have much guidance on it. Typically, what we see required by the guidelines are the fish pathogenicity study and the invertebrate, which is the Daphnia study. And then you can get honeybees, avian, those type of things that would be more an oral dose.
I can speak a little bit to the counting methods though. So like Hank said, I don't think that we have specific experience with an algae study involving a microbe, but we are expanding our ability, automating our cell counting methods for algae studies. And you can, on a case-by-case basis, take advantage of some of the properties of, the differences between algae cells and microbials. So we have a flow cytometer that, counts algae cells based off of a specific fluorescence and autofluorescence. So, if you could identify through pre-test trials that, the algae cell is fluorescent at a certain wavelength and the microbial is not, you may be able to discern between the two.
Right. Let me. We have an early remark as well, and I found it interesting, Hank, that you mentioned getting the right team together, right? This delves into one of the remarks. I think that the exposure should be assessed and considered in tox testing. If the hazard testing is conducted at exposure relevant levels, it would be more meaningful and reduce the physical toxicity, i.e. the gill clogging artifact. Let me bend that toward an actual question. You mentioned the team involved. How are reviewers how do you see reviewers as part of the team that assesses toxicity, right?
Well, to be honest, I'm very impressed with both the Canadian and U.S. People that are on those reviewing teams. And I think the key is that kind of upfront communication with them, because I think they're more than willing to listen, but it's got to be data-based arguments. For example, if you want a realistic environmental concentration, show it. Based on, we did some pre-testing in water and we're finding that, it's not very viable. We don't think it's going to get to water. You might argue, do we have to test? Or we put it in water and it's so opaque, or again, we got gravy. Do we need to, let's tone down and let's get a concentration that's more meaningful, where we might eliminate that clogging of gills or that type of thing. So I would say, what's changed is this is a whole new area. Why the microbial testing has been around for a long time. There's been a few products, again, it's a lot of bacteria and fungi. Bacillus thuringiensis, trichoderma is a fungus. The bacillus is obviously bacteria. We have an understanding of how those go. And there was a lot of work done on those early on. But I think so many more products are coming to market now. It's, to me, a pretty exciting time in agriculture, because it reduces the exposure from conventional chemicals. It has a lot of advantages. But just like conventional chemicals, we didn't, it took some time. It took 15 years to get endocrine off the ground. We're going to have to, I think, have at scientific meetings or that, just more discussions. And I think the agencies or the regulators are open to those kinds of discussions, find a better way.
Right. That delves into another question that potential clients may have. We are trying to get all this from the CRO perspective. you know, when things are data driven, you know, how do you generate enough data to make those arguments and then present that as part of the total package for your client?
Well, I think that has to be some preliminary discussions with the regulators, because now you're asking, how long's a rope, or guess what the EPA or, Canada government are thinking. And so you need to come to some kind of agreement. What data will convince you? What trial should we do? With conventional chemicals in the past, we've done a lot of like water solubility trials. There's a model value, and we've done everything we can to get it into solution. And, we're 10x below that model value. There's just no way. So are you going to agree that if we, do the test at 10x below, is it acceptable? And so, to me, it's about dialogue. And, sometimes it's difficult to get meetings with agencies, but again, I think the microbial groups in the Canadian US government are willing to listen.
Right. We have about 4 minutes left. Can the field of quantitative microbiological risk assessment be mined for improving PEC, EEC modeling for microbial pesticides?
Boy, I don't know if we have the data to do it, or, even the models developed. Back to conventional chemists, there's been a huge amount of model development and improvement in those estimates of EECs. Some ways to do it is actual field measurements in the field. We applied it at X rate. This is what it was, and these were the conditions, and if it was in furrow, it didn't get into the water.
Right.
Those types of things. So, I don't think we're there, and that's where I think that exposure piece we really need to work on.
Right.
And the assessment of exposure, for example.
Yeah, I'm going to bunch a couple of questions together here. And there was a question so far: what is the best method for quantitative analysis of the MCPA? And the other one would be CFU. A question on CFU counting. CFU counting often has large variations, as much as 50% or more, even without contaminations, and much larger than HPLC for conventional chemicals. How do regulatory agencies view that kind of variability and the specs set by the manufacturer?
So I think the CFU count kind of remains the standard method. It's the one that is the most straightforward, and you can actually physically discern between this is the test substance I'm counting, and this is background contamination. As far as the variability, it's just that's what it is in the system. Like, it's a biological organism, and the variability there is always going to be greater than you have for something like HPLC. And I think that's where needing, acceptance criteria that is accepted across the board will become important. So maybe the acceptance criteria is wider. It's a wider range than you get for conventional chemicals, but if you can demonstrate why your variability is high, or it's within that wider range of acceptance criteria, that still leads to consistency across the board.
Yeah. And a quick follow-up to that just came in: are CROs readying themselves for the possibility that confirming identity of CFUs via qPCR may become a requirement?
I know we're preparing ourselves for that.
Mm-hmm.
Yeah. And just to mention with the CFUs and what Sarah alluded to in her talk is part of this is just back to conventional chemistry, that you got good methods in place, and that, what are your acceptance criteria for an acceptable count? How many colony forming units do you need on a plate to be an acceptable read? And do I need to do different dilution factors or that type of thing? And that kind of, setting up methods ahead of time like I said, she mentioned Sanko validation. We have a good understanding of what we're measuring and how it's going to behave in that test system, and need to be able to provide. Over time, I think the CFUs will come down. To be honest, over time, I don't know how much the regulatory agencies have really relied on CFU counts in their assessments, but I think that as we get better and better at it, it's going to be a bigger concern.
Great. One more issue. I know a few questions have come across about availability of the presentation. I do know that Eurofins will follow up with attendees after the presentation for any offline interaction. So that's great. I think that wraps it up for this afternoon. So I'm gonna share my closing screen here for a second and mention that we should thank you for joining this webinar. We extend our gratitude to our two presenters, Hank Krueger and Sarah McCoy from Eurofins Agroscience Services, as well as Eurofins as our sponsor. Our webinar coordinators, Kyle Sawyer, we thank him so much as well, and the webinar committee for their contributions organizing this webinar. Your feedback is available and a follow-up survey may be requested. If you're interested in supporting future webinars and content, please reach out to me. We are planning out 2024, and to give you an idea about what we've done in the past and what holes we might have in the future for that unique presentation. Please visit www.agrodiv.org or the Agro Division YouTube channel. On November 1st at noon Eastern, we'll have a virtual 50th anniversary gala recapping the live events from the San Francisco National Meeting. Putting together this special 90-minute Agro Division webinar will be Dana Barrett from the Agro Division Executive Committee. We welcome all of our Agro Division friends who could not make it to San Francisco. Dana was actually one of those, and those curious about our diverse and dynamic Agro Division. It's a great networking opportunity that's coming up on November 1st. For those that did join us, we also have a few fun and future-looking surprises and Easter eggs that'll complement the fun and friendship of the live meeting. So with that, thank you very much for your participation, everybody, and I hope you have a great day.